Okay, well, welcome everybody. And for those in Europe, those tuning in on a holiday, as always, we're aiming to present frontier research in magnetic resonance and biological systems. And both of the talks today will be recorded, so there's no need for any private recording. And as always, questions and answers can be typed in the Q&A box for the end. So with that, I'll turn it over to the first introducer.
Good afternoon, everyone. So it's a great pleasure to be here, to have the opportunity of introducing Professor Alfonso De Simeone, who is going to give the first lecture today. Alfonso has made a major contribution recently to understanding of the interaction between proteins and lipid membranes. In particular, he has studied the mechanism by which alpha-synuclein interacts with cell membranes, both when it's in its native and also in its aggregated states. So through his research, he has revealed that in the native state, alpha-synuclein binds to synaptic vesicles, in particular to a specific mechanism, which is called double anchor, by which alpha-synuclein can bind in two different vesicles and therefore regulating their clustering and therefore the synaptic transmission. By contrast, when alpha-synuclein is not native and is in some aggregated forms, in particular oligomers, it can interact in a dysregulated manner with lipid membranes and in fact disrupt them and compromise cellular homeostasis. These are really innovative, insightful studies that Alfonso has driven, and he may talk about them today, hopefully. More personally, I've known Alfonso for the last 15 years, and through all the stages of the academic career. First as a mentor when he was a postdoc in Cambridge, and then as a collaborator as he has progressed in his rather remarkable and very fast career. And now he's a professor both at Imperial College in London and at the University of Naples in Italy. So it's a great pleasure, as I said, to introduce Alfonso, and I'm really looking forward to his lecture. So Alfonso, you can take over from me now.
Thanks a lot, Michele, and thanks a lot, the organizers, for inviting me for this talk. Can you see my screen? Okay, good. So today I'm going to show some of our recent results about protein folding and misfolding, and in particular in the context of membrane interaction. So we know that the majority of the proteins that, especially those that are have evolved to be structured in the native state, undertake a conformational transition from the unfolded state to the native conformation, and then this transition is somehow encoded to be efficient by the sequence. However, in some cases and under some pathological conditions, there could be some off-pathway transitions that bring the protein to be trapped into alternative states that are designated as misfolded states. And these states are pretty much known to be responsible for aggregation into fibrillar states of proteins, which typically are called amyloid, depending on the structural properties. And then these states are very important because they are connected to a series of diseases, including Alzheimer's, Parkinson, prion diseases, but also non-neuropathic diseases like diabetes type 2. So if we want to generalize and simplify the pathway of aggregation, we can have a native state that here is shown as a folded state, but it could also be an intrinsically disordered state that eventually would undergo into an amyloidogenic intermediate, an active conformation that is responsible for triggering the aggregation, which occurs first as dimers, trimers, and oligomers, and then these evolve into protofibrils, fibers. And as we know, they are connected to these diseases, but they're also interesting structures for biomaterials, and nature seems to know these amyloids and use in different occasions. Now, the membrane interaction seems to be the center of at least a class of amyloid diseases, because it has been now generally acknowledged that the aggregation brings to oligomers that probably are the most toxic species in many cases, and these oligomers would interact with membranes and disrupt both plasma membrane properties or mitochondrial membranes, lysosomal membranes, and so on. So today I'm going to show a couple of examples of proteins associated with membranes that are involved in these processes of neurodegenerative diseases. The first one is the prion protein, and this protein is a protein that is typically located on the outer surface of the plasma membrane. It's attached to the surface via GPI anchor, and it's a protein that is composed of two domains. There is an intrinsically disordered region, which is in the N-terminus and spans roughly 100 residues, and then a folded region that is composed of 3 helices and a short antiparallel beta sheet. Now, if you look at the chemical shift and you run programs like delta-2D, which gives you probability of a secondary structure, you can see that the helices are pretty much well formed, and the 2 strands that typically you see in crystal structures of prion are also identified, although they don't show to be super stable in terms of chemical shift properties. But also it's nice to see that there are some additional strands that don't come up into the traditional structures, and they probably are transient strands that form and they have the character in the chemical shift that you can see in this type of analysis. Now, the N-terminal region is primarily disordered with some levels of polypolymorph 2 structure in the region that is probably the most amyloidogenic part of the protein. Now, why prion is important? Prion is important for its function, but we don't know yet the exact function of this protein. What we know is that the aggregation into amyloids of this protein is associated with a number of neurodegenerative diseases, collectively known as transmissible spongiform encephalopathies, or prion diseases. And these include fatal diseases like fatal familial insomnia, CJD, but also the mad cow, which was pretty much known in the 80s. Now, the big question is, what's the pathway of conversion of prion from its functional state to this amyloid? And that's probably one of the questions that NMR could be unique technique to answer. So at the moment, there is a general idea that in the native state, at least the globular and structure part of the prion is in equilibrium between a packed structure that typically comes out in the crystal and a structure that is still part of the native state, but it has a minor changes into the packing of the helix 1 with respect to the rest of the protein. Now, this motion seems to be physiological and seems to be prevented to going forward into further misfolding by a series of key gatekeeper interactions, which we identified quite long ago. And the interesting part of these 4 interactions is that all of them are connected to some pathological mutation of prion diseases. So it's one of the hypotheses that these interactions would prevent, this mutation would prevent the interaction to be effective in being an energy barrier to further misfolding of the protein, and somehow lowering the barrier to get into the fibril. So in the work I'm showing today, we study a particular mutant of prion, the T183A, and we chose this one because it seemed to be the most dramatic mutation, which would eventually disrupt a key hydrogen bond that packs the beta sheet onto the helix 2. Now, if you remove the side chain of the threonine, you deplete this hydrogen bond, and we managed to get this system into our NMR tube, into our test tube, which was not very straightforward because it's very unstable as a mutant. And the first thing that we found is that a simple depletion of an hydrogen bond makes a huge difference in terms of thermal stability of the protein. So these are CD melting curves, and the same DSC studies of the stability showed a change in the melting transition of more than 20 degrees. On the other hand, the secondary structure doesn't seem to be specifically altered by this depletion of hydrogen bond. And in fact, the CD properties seem to be very similar, although the mutant had a minor reduction in secondary structure content. Also, if you look at the HSQCs, you can see that the distribution of peaks is pretty much similar, suggesting that the fold of the protein is pretty much conserved by the mutation. And indeed, the location of the main chemical shift perturbation was associated to residues that are close in the space to the mutation side, although some residues in the long range were also identified, which was suggesting some interesting alteration of the protein. So the secondary shift, the secondary structure analyzed by delta 2T indicated that downstream the mutation is here. There is a reduction in helical content in the helix 2, and also in the helix 1 we found a minor reduction in helical content, whereas generally there was a moderated increase in random coil content in the protein. So this indication suggested that the protein is pretty much similar in terms of fold. It is much less stable and has a slight reduction in secondary structure. So what about the dynamics? So we checked a few NMR experiments for dynamics, and we found the CPNG was the one that could capture at the best the alterations in the dynamics of the protein. And in particular, we found that a series of residues were showing relaxation dispersion curves, whereas in the wild type, there is no dispersion at all. So the protein seems to be not interesting from a dynamic point of view in the CPNG timescale of probing. So the residues that showed the alteration in relaxation dispersion curve were all clustered in regions that are across the two different subdomains of the protein. One is the beta sheet, and each one here in red, and the other is the helix H2 and H3. And we pretty much cluster them in three different hotspots. One is around beta sheet, which, by the way, is also broadened. Many of the residues of the beta sheet are broadened beyond detection, meaning that there is a lot of conformational exchange in this area. Another one is in the helix 2, which is downstream the mutation, which is roughly here. And the other one is the helix 1, which is not very close to the mutation site, but eventually it has some alteration in the dynamics. So we wanted to try to understand what's the conformational transition in this mutant. And in order to do that, we combined NMR with advanced simulations. And in particular, we use advanced metadynamic simulations, which is an enhanced technique of sampling, which enables to reconstruct the free energy surfaces in conformational landscapes of protein. So the results show that here is the free energy landscape on a single variable, which is the contacts between the two subdomains that are identified. And then we see that the wild type and also the mutant identify a pretty much defined basin of conformational fluctuation in the native state. But the difference between the wild type and the mutant is that the wild type doesn't go further into changing the packing between the two subdomains, whereas the mutant has a second minimum, which corresponds to conformation where the two subdomains, that is beta sheet helix 1 versus helix 2 and helix 3, are totally detached. So it looks like there is a major native state and a minor populated, let's say, invisible state. And the interesting thing is that the kx that we could calculate from the energy landscape in the simulations were pretty much similar to the ones that we obtained from the CPNG, suggesting that the conformational equilibrium that you see in the CPNG between the native state and this invisible state could be between the crystal structure and this state in which the two subdomains are detached. One interesting thing that we found is that the mutant could generate amyloids, could aggregate very quickly into amyloids. And you can see this from THT fluorescence assays, whereas where the wild type is totally flat, doesn't aggregate, and the mutant aggregates very quickly. These aggregates have the typical signature of amyloids with the 10 angstrom typical diffraction pattern, and also the various times that were showing the morphology of fibers. And the interesting thing is that this is only the C-terminal domain, and there was no observation prior to this one that this domain alone could form amyloids. It was always believed that you need the region 106, 126, which just precedes this domain and is considered to be the amyloid region. So in general, we thought that, so the result that we found is that there is an intrinsic dynamics in the native state, and in the mutant, these dynamics overcomes an energy barrier that brings the population of an active state for aggregation that eventually promotes the fibrillization of the protein. And so we thought, can we identify a way to block this mechanism as a proof of principle? So we accessed a series of antibodies that could eventually lock the protein into the native conformation, and we selected some antibodies that are developed in the Aguzzi lab in Zurich. And I will show just the result with the POM1, which locks the helix 1 on helix 3, so just preventing the motion that we identified. And this was while the mutant, in the absence of the antibody, always aggregates very quickly into fibers. In the presence of the POM1, this was totally prevented. So, so that's an example of misfolding of the protein. Of course, prime is at the surface of membrane. We haven't worked in the presence of membrane in this particular case. But another example of membrane folding and misfolding is the one of alpha-synuclein. Now, synuclein is well known to be the central protein in Parkinson's disease. In terms of structure, the protein is intrinsically dissolved in the cytosol, but it has a nice equilibrium with the membrane-bound state that is reaching alpha helix. And this interaction is likely at the basis of the putative functions of the protein, although again, we don't know exactly what is the function of this protein. But it's also relevant for the oligomers. So the dark side of the protein, these aggregates that in principle are responsible for the toxicity in Parkinson's disease. And it's also relevant to the fibrillization. It was shown by many people that the membrane can catalyze the aggregation of synuclein into fibers, which eventually go into Lewy bodies. And then these are the inclusions that are found in dopaminergic neurons, which don't work effectively in Parkinson's disease. So the way by which synuclein binds membranes and binds everything that can form an amphipathic surface is to a series of imperfect repeats that encode for amphipathic helices that enables the protein to be modular in its binding, so such that it can bind basically everything from a small micelles to planar lipid bilayer. So we have studied this membrane-bound state using solid-state NMR in collaboration with Gianluigi Veglia, and then we found that basically we could identify conditions in which you can study the N-terminal region of the protein. You can see the N-terminal region in cross-polarization experiments. These are typically experiments that tackle transmembrane regions of protein. So that must mean that this region becomes really rigid in the membrane-bound state, whereas the C-terminal region was still highly dynamical and still disordered, to be seen in next experiments in solid state. But they are very similar to solutions experiments, and they typically tackle highly dynamical regions. But however, we couldn't actually see the central region because that's very likely still in conformational exchange between a bound and unbound state, or a helical or tethered state. And for this, we could complement the solid state with the CEST, chemical exchange saturation transfer experiment in solution, which are also in this particular definition known as DES, dark exchange saturation transfer. And basically the idea is that you can look at the unbound state and then run a series of saturation bands at very large offsets. So if there is any interaction with a large object like a membrane, you will see saturation at large offsets, whereas if there is no interaction, like in the C-terminal domain, you see saturation only when the offset is at 0. And in this way, you can follow the profile of binding throughout the whole sequence of the protein, regardless the fact that the central region is broadened in the solid state. And then you can see the N-terminal binding, the C-terminal non-binding, and the central region profile. So using this combined approach, we have scanned the different regions of the protein. We have studied the modes of binding and topology with respect to the membrane or the N-terminal region. We have exploited the properties of the central region, which is quite interesting in terms of both aggregation and membrane interaction. And one of the things, as Michele was introducing, is the identification of this double anchor mechanism in which the protein can bind one membrane with the N-terminus and another membrane with the central region, 65 to 97 roughly, in order to promote vesicle-vesicle interactions. And these are probably at the basis of mechanisms by which synuclein can regulate the flux of vesicles during neurotransmitter release. Whereas the C-terminus was also showing interesting properties with the calcium binding, which seems to be a switch for the interaction. So I wanted to show one of the recent data about this double anchor mechanism, and we were interested into the binding to the plasma membrane. So we studied the interaction of alpha-synuclein with the outer surface of the plasma membrane and inner surface. So in terms of biophysical properties, the lipid composition are very different if you look at the plasma membrane, and there are very different composition and charges in the internal plasma membrane with respect to the external one. And in fact, the external one has very limited binding affinity for alpha-synuclein, very minor interaction properties. However, the internal has a much more stronger affinity for the protein. And the affinity seems to be primarily directed into the N-terminus. In fact, we were surprised to see the type of binding of synuclein with internal plasma membrane, which is primarily all concentrated in the N-terminus, with the central region being almost not interested into interacting with the membrane. And there's a huge difference in the binding property of the central region with respect to the internal plasma membrane if you compare it with the synaptic vesicle binding. So there is a huge difference in affinity. So this suggested that eventually the double anchor mechanism could work even more strongly with the internal plasma membrane, because you bind on the N-terminus and then you have the whole central region available to bind another membrane. So we follow the idea that eventually synuclein can help the stabilization of docking of vesicles with the internal plasma membrane as a competitive mechanism with respect to the clustering of synaptic vesicle, which is another property of synuclein. So basically, we follow the idea that this modified double anchor mechanism could actually be also even more relevant than the one of synaptic vesicle interaction. And this is in line with a series of data, notably the data from the Eliezer lab, where they showed with the exocytosis assays that this could be a possibility. So in order to prove that, we decided to look into TIFF experiments. So we put like a layer of membrane on the surface of glass, and then we imaged the focal plane. You would image just the vesicle, because the vesicle would be fluorescently labeled, and the internal plasma membrane would not be that. So this was the composition of a synaptic vesicle, and this was composition of the internal plasma membrane. So the idea is that does synuclein helps this vesicle to be more in the focal plane? it turns out to be the case. So if you do imaging of TIRF in the absence of synuclein, you see a few events of docking, but in the presence of synuclein, these dockings are much more present, and there is both a concentration dependency of both the number of vesicles that dock into the surface and their residence time. So I will stop here because I think I'm out of time, but just wanted to say that overall, we use this approach to study a series of processes of interaction with membrane into the putative function of synuclein, which is the one of helping the process of regulating the process of synaptic vesicle exocytosis. However, there is also another side of the protein which we are also interested in. I will just show just the idea that it is when the synuclein aggregates into oligomers, which can be originally disordered and then convert into fibrillar species, which are believed to be the toxic responsible objects for toxicity, because they can bind and break the plasma membrane, they can break mitochondrial membrane, and eventually they will evolve into fibers, which now are pretty much very well studied. However, the nature of these oligomers is pretty much a question mark, because they are elusive and difficult to isolate. So we use solid-state NMR to study a couple of oligomers, one toxic and another non-toxic, and this helped us to get a mechanism in which the toxic oligomer will bind to the N-terminus and then use the fibrillar core to break the membrane and therefore generate toxicity. And then we also provide a proof of principle that if you use this structure mechanism to design molecules, in this case it was an antibody selection based on the structure, you can actually stop the toxicity of these aggregates. So with this I conclude and say that PRP has this interesting pathway of subdomain separation to initiate the aggregation, whereas alpha-synuclein has this intimate relationship with membranes that is both interesting for function and for its dark side. And eventually, we can use NMR to guide structure-based drug discovery in the future if we get to know better this type of aggregate species. So I stop here with acknowledging people, in particular Juliana for the alpha-synuclein. For the alpha-synuclein work, and a lot of collaborators, including Michele and Massimo for the PRP work. And thanks for your time.
So, Michele, do you want to read out the questions in the Q&A?
All right. So the first question is from Jeffrey Purslow. So the question is about how the CAPMG relaxation data on the T133 mutant can be acquired reliably, given that this construct seems to form aggregates quickly.
Absolutely, yeah. So we also tried other experiments like CEST, and that was impossible because the signal to noise wasn't actually good. For the prime, we found condition in which we can keep it stable at about 70 micromolar for about 6 hours. You still have a very good signal. So what we did was measuring a couple of CPMG frequencies with one sample, and of course you always measure the reference and then change sample and measure other frequencies. So it was a huge amount of material used in this, and of course the interleaved type of acquisition, such that the different offset were not affected by precipitation of the substrate, but it was very challenging. And in fact, we wanted also to do CEST, but it ended up to be not enough signal for having good CEST profiles.
Okay. So before we move to alpha-synuclein, I had a question about the prion part. So there are hydrogenexane data, which I remember seeing.
Yeah.
Have you looked at those as well?
Yeah, that's a good question. I haven't actually looked in view of this work, but they seem to remember they were Otsu 2001, if I
yeah, certainly that, and there have been probably a few more papers about it. I haven't followed the literature, but
so,
you know,
the one of Otsu, if I remember correctly, they show that the most protected region is the H2H3 around the disulfide bridge. So that would be consistent with the fact that if you get into this intermediate state, you have a pretty much good access to hydrogen exchange throughout the whole S1, H1, S2 domain, plus the flanking region too. So the data would be consistent with that.
Okay.
But I have to look into new ones because yeah, so that's a good point. I haven't actually thought about this recently.
Okay, so there is a couple of questions about alpha-synuclein, so perhaps you can give a short answer if we stay in time.
Yeah.
So the first question is about the assignment of the peaks in toxic and non-toxic oligomers. In particular, yes. You had enough sensitivity for the 3D experiments.
Well, no, we didn't have enough sensitivity. In fact, I mean, the assignment was a combination of indirect data from test experiments, which were giving us the regions where we had roughly the core of the oligomers. Plus, we could get up to the 2D, so we could get like an NCO, NCA data, which helped us to match with the different type of chemical shift. One of the information we had was that the chemical shift of the non-toxic were pretty much random coiled. So we could use to do this assignment or attribution, as Chris Dobson used to call, based on the chemical shift table of random coil, and similarly also for the core of the toxic oligomers we used. It was very useful for us, the structure of, at the time, of Chagrinster, which had all the assignment of the fibula species, and therefore these helped us to identify the resonances based on the combination of these different experiments.
Okay, thanks. So there is another question, I believe, from David Eliza. So this is, you know, I think is a very important question, and I'm sure it is one that you'd like to address. So it's about the specificity of binding to different lipid membranes. So in particular, the difference between synaptic vesicles versus plasma membranes.
Yeah, I mean, I think it's a key point. Hi, David. So I think the major thing is that there are, so synuclein binds to membrane. So and there is the question of avidity versus affinity. So the protein in this particular region, and especially at the active zone in the synaptosome, is probably full of membranes to access, and there is therefore a competition between the different type of binding. So we also speculated that basically the contribution to this process is given by the competition between this type of binding versus synaptic vesicles and the binding to the membrane. Now the point is, in vitro we try to reconstruct conditions to start this binding, but of course we are still far from what's actually happening in the cell, and there are also other binding partners that can also come into play, like calmodulin, that might interact with the N-terminal region, or the chaperones. There is the work of Stefan Hiller and Roland Rieck on Nature last year that showed that the bound state could be also affected by the fact that some HSP protein might sequester synuclein. It's a lot of competition, and there are a lot of factors that come into place, not last the kinetics, because especially during the action potential, I think the thermodynamics becomes a little bit in a secondary aspect, and the kinetics becomes the most relevant one. So it's very difficult to know where is the balance in these competing processes.
Thank you, Alfonso. So there are other questions coming in. I don't know if there is enough time.
Maybe one more.
Okay, so one is about the double anchor mechanism. And in particular, this is a specific binding, but there may be a non-specific binding due to electrostatic interaction. So how do you distinguish between these two modes?
Yeah, so I mean, it's very it's very key to discuss this. I mean, we in our so sort of the way we typically picture these is that you have the two helixes. I think the N-terminal region is now pretty much clear. The conformational specificity of binding in a proper structural state, whereas the central region seems to be now equivalently able to bind in a helical state and in a tetrate electrostatic way. So I believe that probably depending also on the type of lipid composition in the second binding, the second anchor, this could be very well just a tetrate absorbed conformation that is still enough to establish a link. And then there is also the cooperativity effect that you have many alpha-synuclein molecules that could contribute simultaneously to this and making these protein-mediated vesicle-vesicle interaction strong. So yeah, I think both type of models could work, and it's very difficult to know which one is actually working at which time.
All right. I think with this we can close this part, and I would like to thank Alfonso for the really great lecture and very interesting. I have to say this work on synuclein is really have an impact in understanding the function of alpha-synuclein, the dysfunction in Parkinson's disease. So is a very good example of how NMR can have an impact outside the field specifically, but on medicine and drug discovery as well.
So it's a great pleasure to introduce Malene Jensen, my colleague in the Institute of Structural Biology in Grenoble. Malene trained as a chemist at the University of Copenhagen. She did her thesis with the recently departed and much missed Jens Led, where she used paramagnetic relaxation and exchange to study the structure and dynamics of biomolecules. Following her thesis, Malene came to the IBS in Grenoble, where she received an EMBO long-term fellowship to study IDPs, intrinsically disordered proteins, using NMR. She pioneered the use of residual dipolar couplings, paramagnetic relaxation enhancements, and chemical shifts to map the conformational propensities of IDPs, as well as developing exchange techniques to study their binding modes and interaction mechanisms. Following her postdoc, Malene was class first in the National Chemistry Recruitment Commission in 2010 and has had a tenured position since then. She's won a number of prestigious prizes, including the CNRS Bronze Medal, which, contrary to impression, doesn't mean she came third in anything. It's rather an award which is given to the most promising researchers of a particular generation. She's been full research director at the IBS since 2018 and has built up an independent research group at the Institute. She mainly uses state-of-the-art NMR, but combined with other biophysical techniques to study the role of intrinsic disorder in cancer-related signaling pathways. But today, I think, she's actually going to talk about recent unpublished work where she's actually addressing a conundrum that's fascinated NMR spectroscopists since the birth of biomolecular NMR. Without more ado, I'll hand you over to Malene.
Thanks, Martin, for this very nice introduction, and also thanks to the organizers for inviting me to give this seminar. Yeah, so I'm going to be talking about aromatic ring flipping, and as we all know, aromatic residues are extremely important in proteins because they make up a large fraction of the hydrophobic core of folded proteins. And they're really key to protein fold and stability because they can establish a lot of different interactions such as CH pi, cation pi, pi pi, as well as hydrogen bonds. And ring flipping, which is basically a rotation around the Chi2 dihedral angle of 180 degrees. So essentially, there's no difference in the conformation before and after a ring flipping. And of course, the question is, how do we know this occurs? And this was discovered by several groups already in the 70s, where they managed to assign NMR spectra of aromatic rings. And what they could see is that in many cases, a single exchanged averaged NMR signal is observed for the delta 1 and delta 2 protons here, as well as the epsilon 2, epsilon 1 protons, in contrast to two separate signals that you would expect inside a protein in the absence of ring flipping events. So while it's easy to understand the ring flipping can occur on the surface of proteins where you basically have a free rotation, it is more difficult to understand how this can occur in the hydrophobic core of the protein, where usually these aromatic rings, they engage in different types of interactions. And it was also proposed already in the 70s that we may need to invoke some kind of breathing motions in the protein to allow this ring flipping process to take place. In particular, there will have to be some structural reorganization of this local environment to allow this ring to pass through the 90-degree angle. And there's been some beautiful work from many different NMR groups, and my list is by no means exhaustive, where people have been measuring these ring flipping rates as function of temperature and pressure to derive activation energies and volumes, thereby reporting on these breathing motions. But so far, we have very little insight into the structural details of what is actually happening during this ring flipping process. And one of the reasons is that it's very difficult to stabilize a ring flipping transition state or an intermediate where we can find the ring at or close to 90 degrees, and which then would be amenable to structural elucidation. So the model protein that we are using for these studies is an SS3 domain of a MAPK scaffold protein that is called GIP1. It's an unusual SS3 domain because it forms a dimer, where the dimer interface is it covers the region that is usually used to recognize proline-rich sequences. And we started to study the dynamics of this protein, and we quickly realized that at low temperatures, that means below 25 degrees, we start to see line broadening of several of the resonances in the NMR spectrum. And this, of course, indicates that we have some exchange contributions to the transverse relaxation coming from exchange on the microtomillisecond timescale. So in order to study this in more detail, we measured both proton and nitrogen CPMG relaxation dispersion experiments. And here I just report the exchange contribution that we derive from the difference in the effective R2 relaxation rate between high and low, or low and high CPMG frequencies. And as you see, we have exchange in 3 different regions of the protein. And when we look at where this is located in the crystal structure of this dimer, we see that these residues are clustering around tyrosine 526, either within the same monomer or across the dimer interface. So this gave us the idea that this exchange could have something to do with ring flipping of this tyrosine 526. So in order to understand whether this exchange has something to do with this tyrosine, we made a single point mutation, tyrosine to alanine, and we assigned the NMR spectra, and we obtained the exchange contributions again from CPMG relaxation dispersion. And you see here, this single point mutation entirely abolishes the conformational exchange. So as we saw quite a lot of changes here in the NMR spectrum, we of course wondered whether there was a significant structural change that would lead to the disappearance of these exchange contributions. And we solved the crystal structure of this mutant 1.5 angstrom resolution. And as you can see here, the backbone is entirely conserved, and also most of the side chains. And there is only a very local change here at Q520 that slightly moves, which is close to the mutation site. Otherwise, the structure is completely conserved. So this tells us 2 things. So first of all, it tells us that the tyrosine 526 really is the origin of this conformational exchange, but it also tells us that all of the exchange contributions arise from a single exchange process. So this allows us to analyze all of the dispersion data simultaneously on both nitrogen and proton, and we analyze it according to a two-side exchange model where we have a state A in exchange with an excited state, state B, and of course we have to have a chemical shift difference between these two states to observe the relaxation dispersion. So we analyze this according to this two-side exchange model, and we obtain an exchange rate constant between the two states of 2,600 per second, and a population of this excited state of only 3%. So this, of course, this excited state is invisible in the NMR spectra. Here I also report the chemical shift difference between the two states, and you see that for nitrogen it goes up to 5 ppm and up to 1 ppm for proton. So this of course also indicates significant structural changes between the two states. So how can this tyrosine 526 give rise to relaxation dispersion? And of course we cannot be talking about a full ring flipping, which would give a zero chemical shift difference on the surrounding nitrogen and protons. So when we look in the crystal structure, what we see is that this tyrosine 526 is characterized by a Chi2 dihedral angle of 2 degrees. So this means that we have syn-pentane steric interactions between the delta 1 nuclei of the aromatic ring and the protein backbone. This is also illustrated here on the Newman projection, where we have this Chi2 eclipsed structure, where the delta 1 carbon and the carbon alpha are close to each other. And this conformation is, of course, extremely rare. So if you go into the PDB database and you look at tyrosine Chi2 dihedral angles, what you see is that the most common dihedral angle is around 90 degrees, and we are here at 0 degrees. So it's an extremely rare conformation. So how is this conformation stabilized in the structure? And here is the key interactions. So we have a lot of CH-pi interactions from Q520, valine 517, as well as alanine 541. And then there are some pi-pi interactions with histidine 493. And we wanted to try and understand how this specific local environment influences the conformation of tyrosine 526. And in order to do that, we compared our SS3 domain to other human SS3 domains. So we obtained the sequences of 320 human SS3 domains, and we carried out a sequence alignment, and we retained only the SS3 domains that carry a tyrosine or phenylalanine at this position. So this is of course what is similar to this SS3 domain here. And then we tried to look at the amino acid distribution in these 4 positions. So at position 520, 517, 493, and 541. And we assigned a size score to each amino acid according to the number of heavy atoms in the side chains. So this is the distribution you see in these 33 SS3 domains. And as you see in particular in position 541 and position 493, there's really a large distribution of amino acid types and thereby sizes. And we wanted to understand if there is a correlation between these different sizes in these positions and how this influences the conformation of the central tyrosine or phenylalanine. And in order to do this, we carried out a principal component analysis by using as input these size measures that we have here. So the number of heavy atoms in the side chains in these positions. And as you can see, the principal component analysis, I here report the 2 main components. It essentially identifies 2 different groups. And we find the SS3 domain of GIP1 in group 1. And as I also already stated, this the tyrosine here in the central tyrosine is found in a Chi2 eclipse conformation. In group 2, there are already 5 high-resolution crystal structures available in the PDB database. And interestingly, they are all found, the central tyrosine or phenylalanine is found in a Chi2 staggered conformation where we have a rotation of 60 degrees. So at this point, we hypothesized that group 1 SS3 domains correspond to eclipse conformations of the central tyrosine or phenylalanine, and group 2 corresponds to SS3 domains where this tyrosine or phenylalanine is found in a staggered conformation. And in order to support this hypothesis, we identified another MAPK scaffold protein that is called POSH, which has the 4th SS3 domain in group 1 and the 1st SS3 domain in group 2. And we solved their high resolution crystal structures. And again, supporting this hypothesis, you see here, the 4th SS3 domain in group 1 has an eclipse conformation of the corresponding phenylalanine. And the 1st SS3 domain here has a staggered conformation of the corresponding tyrosine. So at this point, we were wondering whether the excited conformation of our SS3 domain G1 is actually a staggered conformation of the corresponding tyrosine. So in order to look into this, we wanted to stabilize the excited state that we detect by relaxation dispersion by introducing single point mutations. And for this, of course, we use the result of the principal component analysis. And what the principal component analysis tells us, we have exactly the characteristics of the surrounding amino acids within the group 1 eclipse conformations or within the group 2 staggered conformations. So here the sizes of the spheres that you see here corresponds to the sizes of the amino acids on the average size, of the amino acids in these positions in these 33 SS3 domains that we were analyzing. And one specific feature that we see here is in position 541, an eclipse conformation appears to be stabilized when we have a very small amino acid in this position. Unlike in the group 2 staggered conformations, this will be stabilized by a larger amino acids in this position. This is exactly in contrast to the position 493, where we typically find large amino acids, in particular aromatic amino acids, when we have an eclipse conformation, and very small amino acids when we have a staggered conformation. Also, there's a difference here at position 541, where in the eclipse conformation, we typically find valine, and in the staggered conformation, we find isoleucine or leucine. So on the basis of this, we designed 4 single point mutations, so alanine 541L, histidine 493A, and V517L. And we added an additional mutation, V517A, that basically was intended to create more space for allowing the staggered conformation to be populated. And we managed to solve the crystal structures of these 4 single point mutations. So here I show you the results. Here at the center, we have the crystal structure of the wild type protein that we already looked at. And when we carry out and we solve the structure of the mutation V517L, there is essentially no difference. So we still find the tyrosine in the eclipse conformation. However, when we go to alanine 541L, we see a transition to a staggered conformation of the tyrosine 526, which is then stabilized by alanine 521, so by CH-pi interactions, as well as CH-pi interactions from the leucine that we introduced. Surprisingly, what we also saw that when we solved the structure of the V517A, we also populate the staggered conformation of the tyrosine. However, the stabilization mechanism is different to the one we found for the alanine 541L mutant. So in this case, this staggered conformation is stabilized by CH-pi interactions from leucine 519. The crystal structure of the histidine 493 alanine variant is identical essentially to the V517A mutation. So again, the leucine 519 is stabilizing a staggered conformation of the tyrosine 526. So of course now the question is if these staggered conformation represents the excited state that we detect in solution. And since we have 2 different structures, so to speak, so we have the alanine 541 structure, but we also have the V517A and the histidine 493A-like structures. I will just focus on the bottom 2 here, and I will show you that this is what captured the structure of the excited state that we detect by relaxation dispersion, but we can of course discuss the other one in the questions. So what are the main structural changes when we go from the eclipse conformation to the staggered conformation? So first of all, we see the main structural changes in the beta strand that encompasses residues 517 to 523. So this is a beta strand that surrounds the aromatic ring. And what we notice is that in the wild type case, we have in this beta strand, we have a classic beta bulge at residue 518. So this is also illustrated in this scheme down here, where the lines here indicate the orientation of the carbonyl. So we have two carbonyls pointing out here. In the mutation in the two mutant structures, we basically have a transition when we go from the eclipse to the staggered conformation. We have a transition from a classic beta bulge to a canonical beta strand, as you see here. And this is exactly what allows the leucine 519, which was previously in the wild type protein solvent exposed, to enter and into the pocket and to stabilize the staggered conformation of the tyrosine in this conformation. So of course, we also wanted to prove that this conformation is what we observe by relaxation dispersion, which is then populated to only 3%. So we did NMR on these two variants. So here you see parts of the HSQC spectrum for two different residues. And what we observe is that we have a linear correlation between the chemical shifts in the wild type protein and in these two variants for a small subset of residues which are unaffected by the mutations. So this suggests that these two variants here are in fast to intermediate exchange between the conformation found in the wild type protein and the conformation found that we managed to crystallize for these two variants. This also indicates that they are not 100% populating the staggered conformation. And in agreement with this, we have extensive relaxation dispersion for both variants. So we recorded these relaxation dispersion data set and we analyzed them. And then we compared the extracted chemical shift differences for the wild type protein and for the two variants. And here I just show the delta chemical shifts for proton. And as you can see, we have a quite nice agreement what was extracted for the wild type protein and what was extracted for the two different variants. So this shows that, that we are able to stabilize the excited state to a significant extent using these two single point mutations. We can, of course, also look into the details of the energy landscape. So what is interesting, if we look at the histidine 493 alanine mutation, we have an exchange rate which is very similar to the wild type protein. And both from the relaxation dispersion as well as the visible shifts here in the HSQCs, that we can see that the staggered conformation is actually only slightly stabilized compared to the wild type protein, yet this is the conformation that we managed to crystallize. For the valine 517A, we see that the exchange rate is much faster than it was in the wild type protein. However, we have a complete inversion of the energy landscape where we managed to stabilize the staggered conformation to 89%. The change here in the exchange rate also indicates that the rate limiting step in the ring flipping is the reorganization of valine 517 that is located just above the ring. Of course, we also wanted to get insight. So now we know that we have a ground state and an excited state, and they correspond to an eclipsed and a staggered conformation. But we also wanted to understand whether we have actually full ring flipping, so the 180-degree rotation. And our carbon HSQCs that you show here at different temperatures, and we have 5 different tyrosines in this SS3 domain. And this, what we see here is basically a single exchanged array signal for all of these different tyrosines, indicating that they all undergo 180-degree ring flipping. And so I will just finish with a movie that illustrates the structural changes associated with the ring flipping of tyrosine 526. So first of all, we see the tyrosine here in the center in the eclipse conformation that is stabilized by multiple CH-pi interactions as well as pi-pi interactions with histidine 493. And then we have the transition to the ring flipping intermediate, where we have a transition from a classic beta bulge to a beta strand. And we have the stabilization of that conformation by CH-pi interactions from the incoming leucine 519. And then we can complete the full ring flip and going back to the main conformation. So with this, I would like to thank all the people that were involved in this work, and it was really a collaborative effort. So the majority of the work on the NMR side and the biochemistry side was done by Laura. And also Luisa was the main driver of determining the structures of the POSH proteins. And there was some input from a former PhD student, Jaka. And there was also some MD simulations that I did not have time to talk about, performed by Nicola and also analyzed together with Martin. And all of this work would really not have been possible without a very nice collaboration with X-ray crystallographers, and they were always very willing to solve all of these different structures. So with the team of Andres Palencia and his postdoctoral fellow, Francesco Ielasi, and we had some nice interactions also with Guillaume Bouvigny at the ENS in Paris. And I would like to thank these sources for funding and you for your attention.
Thanks, Malene. There are already 2 questions in the question and answer. So the ever eagle-eyed David Eliza asks the following question. Just curious, leucine was not one of the favored residues at position 541. Why did you choose A541L?
So I have to admit, when we designed the mutations, they were done on a more limited sequence alignment. So at that point, we didn't do the exhaustive 320 SS3 domains. So it was a smaller subset of SS3 domains that we had at the time. And there, the leucine was one of the more favored ones. But it does appear, of course, in the list. It's not the most favored one. So, but yeah. It's still a larger residues, and it goes along with the size difference between the eclipsed and the staggered conformation.
And there's an anonymous, some are anonymous and some are not. Some are probably not deliberately anonymous. So this one says also, why, this may be David again. Why do you think the histo-ala mutant crystallized in the excited state given the populations you determined? Haha, why do you think it crystallized? I'm looking forward to this answer.
Well, I of course can't really answer that. I mean, maybe it's a more favorable conformation for crystallization, at least under the conditions that that we used. On the other hand, I've been trying to convince the crystallographers for a while to, let's say, look at the conformation. So basically solving the same structure a number of times, and they're not so keen on it, but I managed to at least solve it a second time. So the statistics is already good, and it shows exactly the same conformation. So we had 2 different crystals showing the same conformation. So it must be the crystal packing, at least that is more favorable in these conditions. That is all I can say.
Martin, can I ask a follow-up question?
Please do.
I assume these were crystal structures done at cryogenic temperatures. It
Yes.
seems like it might be useful to look at room temperature crystals.
Of course. I mean, I think we are going to go that way next. Yeah, definitely.
So there's a question from...
Could I follow up on this as well?
Please do.
Did you compare the B factors for the aromatic rings? Because I mean, sometimes people just don't refine against more than one conformation, and I mean, this you may find out by comparing B factors with other more rigid regions in the protein.
Yeah, so we also looked at, I asked this question, and they do not see any evidence of any other conformations in the crystals at cryogenic temperatures, at least. So that's why maybe room temperature would be good. We did look at the B factors, for example, of this beta sheet that appears to be able to rearrange. And indeed, this beta sheet has higher B factors than all the other residues incorporated into beta sheet in the structure. So this appears to be intrinsic dynamic. But this is all I can say. It doesn't appear to be a way to analyze the crystallography data in terms of 2 different conformations, for example.
There's a question from Fleming Hansen. Very interesting talk. Malene, can you confirm the eclipsed conformation of Y526 in solution by, for example, long-range scalar couplings or key NOEs?
Can you go for the eclipse conformation? Yeah, I think we could. Yeah. So we have solved this structure by crystallography in different conditions. So I would say I trust the crystallography in this case because we have the same structure solved under different conditions, so different crystallization conditions of the wild type protein, and it always shows this eclipse conformation. So I think that it's quite certain that it is indeed eclipsed in the wild type protein. But of course, we could go through a proper structure determination to check that in solution. Yeah.
And there's a methodological question or possibly philosophical. So from an anonymous attendee, nice talk. While influence of aromatic is surely agreed upon, how are you sure that it's not, I think, due to Chi-1 motions and only due to Chi-2?
Sure, it's not due to Chi-1 motions. No, I mean, I trust the structures that were determined. So essentially, the only difference between the wild type and the mutant structures is a rotation of the Chi-2 angle. I'm not sure exactly. We did not see any change in the Chi-1 in any of the structures.
Okay, there are 3 more questions. Shall I ask the 3 remaining questions? Is that fine? We're not going over, are we? Or tell me, guys.
Go ahead.
Yeah, go ahead.
Okay, so there's a question from Thomas Sipersky. Great talk. Did you consider measuring aromatic CH-RDCs to validate your 2-state assumption for the flipping?
RDCs, no, but I considered measuring relaxation dispersion on the ring directly. So this is, I didn't think about RDCs, but I was more considering going in the way of relaxation dispersion, using either selective labeling and so on, going in this way to get more insight potentially. But yeah, we could think about that. Yeah.
Indeed. There's a question from Jeffrey Hook. Very nice, Malene. I hope you consider depositing the data, including relaxation dispersion, in the BMRB if it's not already in.
We already deposited all the chemical shifts, but we can, of course, also deposit the relaxation dispersion. Yeah, sure. Yeah.
Goes without saying. And there is a question from Angela Gronenburn. There appears to be no difference in propensity between the Y and F in that position in all the structures. People frequently evoke that there may be a contribution of the hydroxyl. Good question. Any info for a difference between Y and F at this position?
We don't appear to pick up any difference. So this is also why we included both the Y and the F in our analysis of all the SS3 domains. And also when we saw the crystal structures of the POSH, we had one that was a tyrosine and one that was a phenylalanine, and it still appeared to follow the consensus of the principal component analysis. So at the moment, we don't really see a difference, but there could maybe be a difference, of course, with the hydrogen bonding, and maybe we could estimate that a tyrosine at this position could be better stabilized in an eclipse conformation, for example, if there's a possibility for hydrogen bonding. But in what we have seen there, we could not pick up any particular difference for those 2.
Okay, great. Sorry, there is one more question here. Nice talk, Malene, from Himanshu Singh. Nice talk, Malene. Could you comment on the pKa value of this exciting tyrosine?
No, unfortunately not. No, this we have not measured. No.
Okay, I think that's...
Can I ask one more quick one, Mark?
Go ahead, Art. You make the rules.
In the very beginning, you pointed out that the broadening extends across the dimer interface.
Mm-hmm.
You know anything about the dimer in the sparsely populated state?
So actually, in the very beginning of this study, I thought that the dispersion came from a dissociation of the dimer into a monomer, because it is very close to the dimer interface. However, we measured the concentration dependence of the relaxation rates, and we didn't see any differences. we also have a mutant, and this is where it becomes interesting. So we have a mutant that dissociates the dimer into a monomer. And there we have a significant change in the relaxation dispersion. So there's still relaxation dispersion, but it's clear that the time scale is changed and it goes towards a slower exchange rate. So because it's slower exchange, we have very significantly less exchange contributions. And I think that really this rearrangement is only possible because it, to some extent, occurs at the dimer interface. So in the monomer, this occurs on a different timescale, that's for sure.